Hello, my name is Matthew and I'll be presenting the project by me, Remy, and Howie about meiosis. For meiosis to begin, it must go through interphase. The first step of this process is cell growth. During cell growth, a cell membrane is formed with a nucleus that has chromatids inside. Once the first growth is complete, DNA synthesis begins. This is the process of chromatids copying each other. Each homologue attaches to its copied sister chromatid at a centromere. After this, there is more growth, and then it prepares for division. The formed chromosome begins to condense and become visible. The process of two chromosomes forming is called synopsis. Next is prophase 1. During this phase, the two homologous chromosomes stick together in pairs to form something called a tetrad. The chromosomes then exchange parts called a chiasma, then with each other during a process called crossing over. After this is done, the nuclear envelope begins to break down, and the centrosomes start moving towards opposite poles of the cell. The spindle fibers begin to form, beginning a process called spindle apparatus. The spindle fibers begin to try to connect the kinetic core. When the spindle fibers attaches, it is called a kinetic core microtubule. After this, the process of metaphase 1 begins. The tetrads begin to move towards the middle called the metaphase plate. They then line up and attach to microtubules so the anaphase process can begin. During anaphase 1, the chromosomes are pulled apart by the microtubules to the opposite poles of the cell. The cell begins to get stretched apart by the force of the chromosomes. The microtubules are then removed again so the next phase can begin. Next phase is telophase 1, where two separate nuclear envelopes form. Cytokinesis then occurs to split the cell into two cells. The divider in between them is called the cleavage furrow. After cytokinesis and meiosis 1, oogenesis produces one large haploid secondary oocyte and a small haploid polar body. The oocyte keeps most of the cytoplasm, and it begins meiosis 2, but pauses until fertilization occurs. Once this is complete, now meiosis 2 can begin with prophase 2, where another centrosome appears in each cell. There are now two centrosomes in each cell, and the nuclear envelope breaks down again. Then a mitotic spindle begins to form to attach chromosomes at the centromere. Metaphase 2, chromosomes move to the center and line up at the metaphase plate. As microtubules attach to each sister chromatid in anaphase 2, the sister chromatids are pulled apart to opposite sides of the cell. During telophase 2, the nuclear envelopes form. Microtubules disappear and cytokinesis 2 splits the cells. After this is spermogenesis. 4 haploid spermatids are formed. There are no mature yet. Spermiogenesis begins when the developed tails condense their DNA into a head from a chromosome and they lose extra cytoplasm. This is our explanation of meiosis. However, there is still one similar phase that must be mentioned, which is mitosis. Mitosis is the splitting of a cell to create an identical cell. Unlike meiosis, it only happens once, and its interphase is the same with G1, S, and G2. With it only cycling once, it only has prophase, metaphase, anaphase, telophase, and cytokinesis. These phases are pretty similar to mitosis 1. Also, mitosis happens during asexual reproduction, so there is usually no variation between cells, unlike meiosis. Thank you for listening. This is Matthew signing out. And yeah.